Welcome to AQA Combined Science, Paper 2, 2023, Trilogy. Let's get to the marks. This paper takes an hour and 15 minutes. It's for the higher tier. Try to pause each question before you have a go, and then watch the walkthrough for the question. Let's go. Okay, so question 1.1. Name the type of cell division that produces egg and sperm cells. You've got two types of cell division you need to know about. This is mitosis and meiosis. So which is which? Well, meiosis contains the letter E, and I think of that as E for eggs, and it contains the letter S, and I think of that as S for sperm. So meiosis is used when we need to make eggs and sperm. Mitosis is for asexual reproduction, and it's for growth and repair. And so that's not the correct answer here. Correct answer is meiosis. Okay, let's look at 1.2. Sexual reproduction produces offspring that are genetically different from each other. Give two reasons why sexual reproduction causes variation, that means differences in the offspring. So why don't you look like your brother or sister? Why is it that three children don't look alike? Obviously, unless they're twins. So what we're looking at is one of the reasons is when fertilization takes place, you have an egg, and a woman has about 2 million eggs. So who knows which one she's going to release? That's random. And in about a teaspoon of sperm, there's 200 million. So we have no idea which sperm is going to join with which egg. We can say that the process of fertilization is random. We don't know which sperm is going to join with which egg. And of course, we should remember that each of the sperm are different than the other ones. And each of the 2 million eggs in a woman's ovaries are different. So that point's going to score us 1 mark. Now let's look at some other marks that we could get. Of course, when the sperm and the egg meet, what happens when parents create sperm and egg? They halve the amount of chromosomes and then blend them together when they make a new cell, a daughter cell, called the zygote. So this means that the embryo will be made up of genes from the mother and genes from the father. So fertilization involves the mixing of genes, or sexual reproduction involves the mixing of genes from both parents. And this only happens in sexual reproduction. In asexual reproduction, identical genes are passed to the offspring. Okay, let's move on to 1.3. So, polydactyl is an inherited disorder. The allele for polydactyl is dominant D, shown by the capital letter D. A person with two copies of the allele, lowercase d, will not have polydactyl. A person with the genotype big D is homozygous. So, what word describes the genotype big D, little d? And of course, that word is heterozygous. Homo means the same. So lowercase d is homozygous recessive, and big D is homozygous dominant. A person with the genotype big D and a person with the genotype little d have plan to have a child. Determine the probability that the child will have polydactyl. You should complete a Punnett square diagram. Identify any offspring genotype that would have polydactyl. So here we put one of the parents' genotypes, big D, little d, and on the side we put little d for the other parent, and then it's a case of just cross-referencing them together. So we get big D, little d, little d, little d, big D, little d, little d, little d. Why is it 5 marks? I have no idea. These questions are normally about 3 or 4 marks unless there's an extra complexity, like saying is it a male or female child, but let's see. These ones have polydactyl. You could circle it and then label it to show that anything with the big D has polydactyl. So, yeah, that's the genotype that would have polydactyl. The two little d's are homozygous recessive and would not have polydactyl. So now we need to find out, well, we've got 2 out of 4 with polydactyl. So there's a 50% chance, 2 out of 4, or 0.5 probability. And that shows we have polydactyl. There we go. That's 5 marks. It seems a bit easy. They were generous that year. Once again, pause the video before the questions. 1.5 embryos can be screened, that means checked out, for alleles that cause inherited disorders. What are two advantages of embryo screening? Well, if you're a parent and you find out that your child may have a genetic disorder, you can make preparations before the baby is born. So you can be prepared to seek medical advice, do your own research, find out what things you might need to help that baby growing up into an adult. Also, it may be an advantage. The parents may choose not to have the baby as they're planning to have a baby, and this may lower the amount of people with the disorder in the future. They may make the decision that they couldn't handle having a baby with extra needs and therefore plan not to have the baby or maybe find a different partner, which might be drastic, but still. Question 2.1. Why is the percentage of species that are extinct only an estimate? So, the amount of its species that are extinct in the world, why is it only an estimate? And the answer is extinction. Well, it's not always caused by humans. We know meteors can cause extinction or competition between predators and new diseases. The answer is that humans have not found evidence of every species. And we all know that all individuals of one species do not have the same genes as most species reproduce sexually, but even then there are mutations, even in asexual reproducing organisms. What evidence is used to study species that have become extinct? Well, we can use fossil evidence, and we can also use DNA evidence, or even look at tissue that's been recovered. Okay, let's move on to the next question, which is going to be 2.3. Let's just get that in shot. A bacterium called Clostridioides difficile, let's just call it C. difficile, thank you AQA, can infect the human digestive system. C. difficile can multiply and produce toxins. Of course, we know bacteria produce toxins. The toxins cause diarrhea. Doctors are concerned that new strains of C. difficile may evolve. Antibiotics may not be able to kill these new strains. Explain how the evolution of antibiotic resistant C. difficile can be slowed down. So, this question is specifically asking us how the evolution of antibiotic resistant bacteria, C. difficile, can be slowed. Because it's obviously a bad thing if they become resistant to antibiotics. Antibiotics are medicines which actually kill bacteria. Well, first of all, let's address how, how bacteria become antibiotic resistant. This is something that could be asked in an exam. So check out my answer here. I've spent half of it explaining how bacteria can become antibiotic resistant. You get new strains when mutations cause some bacteria to have a resistant gene to the antibiotic. If exposed to the antibiotic, the bacteria with the resistant gene may survive. They reproduce rapidly, producing a large population of bacteria with the resistant gene in a process called natural selection. Unfortunately, all of this is irrelevant to the question because the question specifically says how can the evolution be slowed down of the resistant strains. It doesn't say how can you explain how an antibiotic resistant strain is formed, which is what I've said here circled in the red. So what you really need to focus on is things like antibiotic resistant bacteria, the production of them or the evolution of them can be slowed down by only prescribing antibiotics for bacterial infections, as antibiotics won't kill other pathogens like viral infections. So the correct precise prescription of them. Also, do not prescribe antibiotics with mild infections, which the immune system can deal with. Also, patients must finish the course of antibiotics so bacteria resistant strains do not survive. Then there's good practice like washing hands, good hygiene before eating, cleaning surfaces with antimicrobials or antiseptics, isolate people with diarrhea or other symptoms so they cannot spread the disease. And all of these kind of things would get you the 6 marks. So this question was mainly focused on how can we slow down the evolution? We can also prevent the spread by herd immunity. That's by creating a vaccine for C. difficile. And of course, you know vaccines, we use dead or attenuated, weakened pathogens in an injection, and that gives people their immune system the ability to make the memory cells that make the correct antibody to destroy the disease. You wouldn't need to explain all of that. You just need to explain the processes that need to take place in order to slow down the evolution. And yeah. So that's, I'm going to put this all together. Let me get rid of the irrelevant stuff. And so there you go. There's your answer, which would score you at least 6 marks. Plenty of information on slowing down the evolution of resistant strains of bacteria and what we can do to slow down the evolution of these resistant strains. Okay, let's move on to the next question. So if we look at 3.1, figure 1 shows a river next to a field. 3.1, describe a method to investigate how the distance from the river affects the number of different plant species in the field. You should explain how to use a transect As usual, I recommend good exam practices to highlight or underline keywords in the question, in the stem of what we call the stem of the question, the instructions, so you don't make any, you don't forget. So I'm going to draw a little diagram and then I'll go through the answer. So we've got the river here, we've got the field. A transect is where you put a rope or a measuring tape along the area you want to measure. And then at regular intervals, you place one of those square metal frames. You probably can remember what they are. They're called quadrats. So at regular intervals, you place quadrats. And then you're going to look inside the quadrat and count up all the different species. You need to identify the different species of plant and then count them up. And then you record them and maybe do that in a kite diagram, which is a special type of diagram that's very useful for showing distribution with transects. The only bit that you need to randomize here, because you definitely don't want to say to place the quadrats randomly. You do that when investigating population size, not distribution. The only bit you want to say randomizing here is perhaps randomize the start place along the river. So to avoid bias, you don't get to pick where to start along the river. And you probably need to repeat that in several places. So let's get this written up. Place a rope or tape measure. I think it's better to use a tape measure because it has the distance on it. If you use a rope, you'll need a meter stick or a tape measure anyway to place your quadrats in regular intervals. Randomly choose a starting place. You can do that using a number generator on your calculator. Place a quadrat at regular intervals. That could be every meter, every 50 centimeters, or 0.5 meters, or every 2 meters, which I've written here. Place that next to the rope or tape measure from starting at the river to one end of the field, or you could start at the field to the river. It doesn't really matter. Then look in your quadrat and identify the different species or plant species in the quadrat. And now you can either count them up, count the number of species in the quadrat, or you can estimate percentage cover depending on the species. Okay, so things like grass is too hard to count because they're so small, or moss. So, well, moss aren't plants, but grass is a plant and it's too small to count. So, um, it's best to estimate percentage cover with something like grass. And use your mobile phone or use a key to help you identify. A key is a card with some photographs of different plants on it and some other information to help you identify the plants. Or you could just use Google Images on your mobile phone or various apps now they have on mobile phones that can identify plants if you take a photo of them. And then plot a kite diagram. The only thing probably to add here is that we should repeat 3 times at maybe 3 other locations or 4 other locations along the river. And then what we can do is take a mean for each set distance from the river. We can calculate a mean distribution. And there we go. Okay, so this turned out to be a 4 marker, so we put plenty of points there and we would easily pick up all 4 marks here with all these points. Sometimes these are 6 markers. Just watch out for those quadrat ones that say population because that is a totally different method. That's where you randomly throw the quadrats or place the quadrats using a grid reference. All right, let's crack on to the next question. Students used a valid method to investigate how the distance from the river affects the number of different plant species in the field. So this means it was valid. It means they controlled all the variables that could affect the number of plant species other than distance from the river, which is their independent variable on the x-axis. And the question says, what is shown by the data in figure 2? Tick one box. Fewer different species are always recorded nearer to the river. The mean value of students can be most certain about is 5 meters from the river. The number of species recorded 6 meters from the river is anomalous. Well, look at the size of these lines. These are called error bars, and they show the deviation or the range around the mean. So what we can find is the mean value of students can be most certain about is 5 meters from the river. Well, look at the one at 5 meters. It's got a really small line, and that means the maximum value and the minimum value above and below the mean are smaller. So our results were more closely grouped together here. This is a more precise or a more accurate and more reliable result, as it has a smaller range around the mean. Okay, let's move on. Cows walk on the ground near the river more than they walk on the ground further from the river. What is an abiotic factor that could affect the number of different plant species found near the river? Well, first of all, the cows walking on the ground. Cows are a living thing and they trample plants when they walk on the ground. They are a biotic factor that is affecting the distribution. Okay. What's an abiotic factor? Abiotic means not biotic, so it means not living. Abiotic is non living. So we need to look at these answers below and first of all see which ones are living. Microorganisms near the roots. Well, microorganisms are biotic, they're living things. Moisture levels in the soil, that's abiotic. Then we have the oxygen concentration in the air. Oxygen's not alive, it's an element or a molecule, so it's abiotic. And then we have primary consumers in the field, which is biotic. So we're really looking at answer 2 and 3. And the answer is 2, the moisture levels in the soil. And why is that? Well, oxygen in the air, it's very rarely is that restricted enough to affect living organisms. It makes up 20% of the air roughly. And also, oxygen is not a limiting factor for photosynthesis like moisture levels, which you would expect moisture levels to affect the growth of plants and maybe the distribution. In fact, of course, we know oxygen is actually a waste product of photosynthesis and so not likely to affect plant distribution in these circumstances. 3.4 Increasing numbers of cows are being farmed across the world. Explain the environmental implications of the increasing number of cows. So, first of all, let's look at how having lots of cows have environmental implications. Well, more cows means they're going to trample up the plants and kill a lot of plants. But they also might eat the plants because cows are herbivores and they like to munch on plants, obviously. What does this mean? Well, this means if we have less plants in the world because the cows are trampling them all and eating them all, we're going to have less photosynthesis, which means less carbon dioxide, is removed from the atmosphere. This means we're going to get an increase in carbon dioxide levels in the atmosphere. Let's also not forget that more cows means more methane. Basically, cows fart out methane. And of course, they respire as well. They're living things, so they release CO2 into the atmosphere as well. And there'll also be more forests cleared for farmlands. Why do they do that? Well, farmers need to keep the cows somewhere, so they have to create fields. And in order to do that, they cut down trees. And all of these problems increase the amount of greenhouse gases in the air. So the increased CO2 and methane, which are greenhouse gases, cause global warming. And global warming causes climate change. Okay, so the planet's warming by the greenhouse effect, and this is causing climate change. And of course, that can lead to floods, extinction of species, loss of habitat, and a change in distribution of species, as well as many other consequences. And this should definitely get you all 6 marks. 4. Sea lice are small animals that feed on the skin and blood of salmon fish. Figure 3 shows sea lice attached to the skin of salmon. Oh my gosh, I can't say that word. What is that word? Oncorhynchus or Kincus, whatever. That's known as the binomial name. And we know that because it's written in italics, that weird slanted words. Now, the genus is always the first word in a binomial name, and the second word, Kita, would be the species. So the answer is that first word written up there in italics. Now the 4.2 says which domain are seed lice in? So what are they classified in? Well, there's three domains. You've got eukaryota, I just call them eukaryotes. You've got bacteria, and you've got archaea, which are the ancient thermophilic bacteria that lived near volcanoes. So are there any clues in the stem of the question? Well, look above. It says sea lice are small animals. And that means they must be eukaryotes, because all animals are eukaryotes. All animals have a nucleus. Okay, 4.3. Some salmon have genes that result in fewer sea lice attaching to the skin. Describe how fish farmers can selectively breed salmon that sea lice cannot attach to. So basically, you're going to choose a male and a female salmon with the gene for reducing sea lice. And you'll be able to spot these because they'll be the salmon that have the least sea lice attached. So you can observe it with your own eyes. You then take these male and female salmon and you breed them. And hopefully these salmon will produce some of their offspring who also don't have a lot of sea lice attached, suggesting that they have that gene, that beneficial gene, which gives them the beneficial characteristic. Now, you can't just do it once. You've got to then take those offspring and breed them again, and take their offspring that don't have sea lice attached and breed them again, and keep doing it until you produce a variety of salmon that, or a group of salmon that do not have any sea lice attached. Then you've been successful. Now, this might take many generations, maybe 5 or 6 or more generations to achieve. So we really just want to highlight, I can't spell offspring. What a Wally. Never mind. Right. So we just really want to highlight the key main points are that you have a male and female salmon, that you breed them, you choose the ones which have the least sea lice attached, you breed them, and then you take their offspring, you select the offspring with the least sea lice attached, and you breed them again. And then you take their kids and you breed them again, but always choosing the ones with no sea lice attached. Okay, let's move on to 4.4. Explain the advantages to salmon farmers of producing salmon that do not have sea lice attached. Well, what is it that these sea lice do? They're going to feed on the blood of the salmon. And this is going to mean that if you don't have sea lice attached, those salmon will be able to grow bigger. They don't have to use their immune system to fight these lice off, and they have more energy for growth. What does that mean? That means less money spent on food to feed the salmon, or that if you give them the same amount of food, these salmon will grow bigger because they can spend more of their energy on growth. And for the farmer, this means they can be sold for a greater profit and make more money. And also, more salmon survive, so you get bigger profit. Basically, less infections, less spread of sea lice throughout your whole kind of group of sea lice that you're farming. Okay, let's move off. Okay, 4.5. Explain the disadvantage of selectively breeding salmon. Don't refer to cost or time. So don't say, oh, it takes a long time, many generations. The actual disadvantage is that when you do selective breeding, you end up with a reduction in the gene pool because you're excluding certain salmon because they don't have the gene you want, which means you're going to lose some of the genes in that population. So you're also reducing the biodiversity. This can mean that the salmon have less genes in that population and so are more susceptible to the spread of a new disease or a sudden change in the environment. If the global temperature changes, maybe we lost a gene that would have helped them adapt to hot climates. So less genes and less variety means there are less characteristics which make them able to adapt to change, such as a new disease. Um, Genes that are lost may also be used by humans in the future for medicines or other things. They might be very useful resources, those genes. So it's like if we did the same thing with the willow tree and we never got the gene that produces the medicine that we now know as aspirin. Okay, let's move on. 5.1. Which hormones are given to women having in vitro fertilization treatment or IVF treatment? Tick one box. Well, we've got FSH and LH, that's follicle stimulating hormone and luteinizing hormone. FSH and estrogen or LH and estrogen. And the answer is the top one, FSH and LH. FSH or follicle stimulating hormone matures, ripens, or develops eggs in the ovary. And the LH, luteinizing hormone, releases eggs from the ovary in ovulation around the 14th day. If you give a woman these, then they're going to produce more eggs and they're going to release more eggs. Right, name the target organ of the hormones used for IVF. Well, this is my crusty little D grade GCSE drawing, more like a G grade, being a bit generous. The pituitary gland produces and releases FSH and LH, and they travel through the blood down to the ovaries where they act and ripen the eggs, and then LH releases the eggs. So the answer to this is the ovaries. 5.3. Describe why microscopes are needed in the process of in vitro fertilization or IVF. The idea is that you basically examine the embryo and ensure that fertilization has occurred properly, i.e., you can also study the quality of the sperm or the eggs before you fertilize them. You can examine the quality of the embryo and make sure it's one that you believe will be fertilized. Don't check out my spelling of embryo, but that's essentially what you're doing, looking at the sperm, the eggs, and the embryo and making sure they're of high quality. 5.4. Describe how the hormones given to women during IVF treatment interact with other hormones to prepare the body for pregnancy. Well, FSH, otherwise known as follicle stimulating hormone, stimulates the release of estrogen. Estrogen develops the lining of the uterus. Now, it's not on the mark scheme, but estrogen also reacts by or works in a negative feedback mechanism to inhibit further release of FSH so we don't waste any more development of eggs. But specifically here, we need to know that FSH releases estrogen, which develops the lining of the uterus. Now, estrogen stimulates the release of luteinizing hormone, which is LH, and luteinizing hormone is what causes ovulation, the release of the egg on the 14th day. And LH stimulates the release of progesterone, which maintains the lining of the uterus. So we have this interaction between the four hormones. Okay, let's move on. That will get you your 3 marks. So let's move on to 5.5. Scientists studied women who had IVF treatment. Table 1 shows the results. We have received IVF treatment. 450 women got treatment, and 135 were successful with their treatment. This question says, calculate the simplest ratio of the number of women who had successful IVF to the number of women who were unsuccessful. So we need to give the ratio in whole numbers. We need to calculate the simplest ratio and then give it in whole numbers. So we have 135 that were successful. 450 was the total. So we need to do 450 minus 135 to find out that 315 was the number of unsuccessful women. So we now need to ratio 135 to 315. And the best way to always do this is to divide both by the smallest possible answer. So if we do 135 by 135, we get 1. And if we do 315 by 135, we get 2.33. Now you've got to think we've got to turn these into whole numbers. It is a ratio of 1 to 2.33. What can we multiply them by to get them near enough into whole numbers? Well, if I multiply 2.33 by 3, I'll get 6.99, which is roughly 7. And if I multiply 1 by 3, I'll get 3. You have to multiply both by the same number. And so we get a ratio of 3 to 7, which is the simplest whole number ratio. Now let's look at 5.6. So just one factor that can affect the probability of a woman having a child as a result of IVF treatment. So obviously the amount of eggs she produces will be important, and the quality of those eggs, and they can be affected by age. The quality of the eggs go down rapidly with age, obesity, and obviously the number of eggs that can be collected from the IVF. These are all factors that would score you a mark. Give 2 arguments against the use of IVF treatment. Do not refer to cost or religion in your answer. So IVF can be very stressful. It can cause strain in relationships, involves a lot of hormone treatment. So psychological problems, mental health can be an issue. It's not always successful. Again, that can contribute to mental health, and it can be a long, physically tiring process. Okay, 6.1. Let's have a little look. What have we got? Give one internal condition controlled by homeostasis. Other internal condition, do not refer to temperature. So the answer there is either going to be glucose or water levels. And of course, that's specifically blood glucose levels or the water level of the blood. There are many other conditions that are controlled, but for GCSE, these are two of the ones you should know. 6.2, explain why the control of body temperature is important. Okay, think about it. What happens if your temperature gets too high? So what happens to enzymes in your body which control all of the chemical reactions in your body? Well, here's a diagram of an enzyme. And when their temperature gets too high, they become denatured and the substrate will no longer bind to the active site of the enzyme. So again, enzymes could denature if we don't control the temperature. On the flip side of that, if the temperature is too low, then the kinetic energy of the enzymes will be too low and there'll be less collisions or less frequent collisions between the substrate and the active site of the enzyme. And metabolic reactions may happen too slowly for life. So either way, too high or too low is not good. And it's very important that we control that. Moving on to 6.3, we're going to look at a little graph here. Figure 4 shows how the body temperature of one swimmer changed in the first 4 hours of a long distance swim. If we look at the y-axis, which has body temperature, we can see over time it's decreasing. You can see the time on the x-axis, and over time the temperature of the swimmer is decreasing. So let's look at the question. 6.3, calculate the mean rate of body temperature decrease per hour in the first 4 hours of the swim for 2 marks. So we want decreasing degrees per hour over the first 4 hours. So what we want to do is mark out 37 here. We then want to draw a line here to the lowest point, and we find that that's 35.5. So we want to do 37, take away 35.5, and that gives us a total time of, a total temperature decrease of 1.5 degrees. So that's the drop on the y-axis in the body temperature. Now, when you're doing rate, you're always dividing something by time. And here we've got hours along the bottom. So we want to do 1.5 over 4 hours. And the answer we get is 0.375 hours, 0.375 degrees per hour. Okay, 6.4. Hypothermia is a dangerously low body temperature. For this swimmer, a 5.5% decrease in body temperature from the start of the swim will cause hypothermia. Determine the body temperature at which the swimmer will start to have hypothermia. Give your answer to 2 significant figures. Always highlight and underline keywords in a question. It'll help you. So, their body temperature at the start is 37 degrees. We know that from the graph above. Let's work out what 1% of 37 is. So if we do 37 divided by 100, we're going to work out that 1% of 37 is 0.37, and that's 0.37 degrees is worth 1%. Now we know that the body temperature dropped 5.5%, so we do 5.5 times 0.37, and we'll work out how many degrees 5.5% is. And that equals 2.035 degrees. I could have also times 37 degrees by 0.05 to work out what 5% is, and I'd have got the same answer, 2.035 degrees. Now, if we do 37 degrees, take away 2.035 degrees, we'll find the temperature that is a 5.5% drop. So 37 minus 2.035 degrees is going to work out to be 34.965 degrees. And that's the temperature at which hypothermia will set in. But we've been told to round this to 2 significant figures. Now, because the 9.6 is bigger than 5 and it makes it closer to 35, we round to 3.5. There are 2 significant figures. 35 degrees is the answer. Okay, let's look at 6.5. Which gland secretes hormones to stimulate the adrenal glands? Well, hormones that stimulate the adrenal glands is known as the thyroid gland, and it releases thyroxine, which acts on the adrenal glands and helps speed up metabolism. Explain the role of the adrenal glands in responding to a decrease in body temperature. Well, the adrenal gland releases the hormone adrenaline, and that will get you one of the 5 marks. The hormone adrenaline increases your heart rate and makes it beat faster. So this increase in heart rate is going to have a greater delivery of oxygen, and don't forget glucose, to the muscles. If you have more glucose and oxygen, what can you do more of? Well, the muscle cells or the muscle tissue, which is a group of cells, can do more aerobic respiration and release a lot more energy. And what do muscles use that energy for? They use it for contraction. And we contract our muscles when we shiver, and so therefore release heat and warm up. Respiration releases heat, and that helps us warm up, or shivering releases heat as well. Easy 5 marks here, so let's move on to the next question. Here we go, the final question. 6.7. Explain how the thyroid gland controls the response to a decrease in body temperature by negative feedback. Well, the thyroid gland releases that hormone called thyroxine. Now, thyroxine does stimulate the release of adrenaline from the adrenal gland, but it also increases your general metabolism, increases the rate of respiration. And what this does is this increases the body temperature. And then the increase in the body temperature negatively feeds back and inhibits the release of thyroxine. And there you go, it's controlled. So the thyroid gland releases the thyroxine that informs the metabolism to speed up. And then that causes a release of more heat because there's more respiration. And that increases the body temperature. And then the thyroid gland responds to the increase in body temperature by reducing the amount of thyroxine it releases. And we call that process negative feedback.
You're still here? It's over.